Welcome back. We will do this question over here, which you can see now. It shows the female and male flower. Obviously, if you just see the anther, this is a male. Okay, this is a male, and this is a female, because this has stigma. So now, with reference to figure 4.1, suggest how the flowering habit of maize encourages wind pollination. So flowering habitat, I guess. I mean, what thing can we say that encourages wind pollination? So they're long anther. Okay, they're long anther. There is an open exposed stigma. So that's the point which I can think of. Let's see what the marking scheme has. There is anther loosely attached at one point to filaments. So we can see here. Okay, these are the anthers. So the first thing we will have here. So this one will be very, very thick. Maybe we can use just this simple line. Okay, and we can say that we can also make this thin. We can say that anther, or maybe we can use this ink. Anther loosely attached, right, to filament. Right. I guess it can be seen from the diagram here that anther is actually loosely attached to the filament. And then we go to the next question where it says, and the next point it says anther on long filaments hangs out of plant. Okay. So we can also see that this is the first point about anther. You can see it is loosely attached, and then we can also see that the anther is actually, you can see that this anther, now we can see that this anther is yeah. So the second point will be anther is loosely what did we read over there? Okay, so as you can see here, anther is loosely attached, the first point, and here we can see that attached to a long filament outside the plant. Okay, it's almost outside the plant. The plant is over here. This is very far away. Okay, and there is a long filament, as you can see over here. So hope I made that point clear as well. Then we have anther stamen high up above leaves. Okay, as we can see, the anthers are very high above leaves. We can't see the detailed version of this question. I mean, here we can't zoom in, but then normally in this kind of things, I mean in this the anther is normally, as we have been told, okay, above the leaves. Okay, is above the leaves. So these are the 3 points about anther. Okay, that's the anthers. And then we have stigmas. Stigmas hang out, and stigmas has large surface area. Okay, so we can see over here there are, in the third point here it was about petal access to wind, no petals. Okay, so stigmas have 3 features. Okay, it's large surface area and it's hanging out. So stigma, about stigma we can say the following. About stigma. Stigma, okay. First, it is hanged out completely, okay. It's hanged out. As you can see, it's out. You can also say that stigma is having large surface area, and finally you can say that stigma, this is the 2 points, okay, about stigma. It's hanged out and it has large surface area. And then there is another thing about this whole plant, okay. If you have noticed both the plants, they do not have no petals, okay. There are no petals. So also this is an adaptation sort of, okay. There are no petals. You cannot see any petal over here. So this helps the pollen grain to come and hit the stigma, okay, without hitting the pollen grains. So that's how the maize plants are adapted. Now we go maize plants, sorry, they are adapted that way. Then we have in a maize plant, the anthers normally ripen and release pollen before the stigmas are mature, okay. Anthers ripen and release pollen grain before the stigmas are mature and ready to receive pollen. This encourages cross-pollination. Spend two potential advantage of cross-pollination to a plant species. This is the point which you already mentioned. It increases it increases hybrid vigor. Okay, we said about hybrid vigor because there is more heterozygosity, okay. There is genetic variation. So genetic variation, okay, it is more adapted. Genetic variation, more adaptation, can be adapted more to the environment and all that. The points are already given over here. Not here. Here we have hybrid vigor, variation, adapt to the environment more precisely. Harmful recessive allele won't be expressed, and then heterozygosity, okay. So hope we remember that. And the next question is interesting one. We have our wheat and maize are grown affect their ability to photosynthesize, okay. The conditions in which wheat and maize are grown affect their ability to photosynthesize. Figure 4.2 compares the rate of photosynthesis of wheat and maize at different temperatures. With reference to figure 4.2, compare the effect of temperature on the rate of photosynthesis of wheat and maize. Okay, now we can, this is a very simple question actually. You have your wheat, you have your maize. So you have your 2 marks. The first point you should mention is that the rate of photosynthesis of wheat is always lower than the rate of photosynthesis of maize. So maize always higher than wheat. That's the first point. And then the second point is you should write that after reaching this temperature of around 18 degrees, the wheat the wheat starts, the rate of photosynthesis of wheat declines, whereas the rate of photosynthesis, the plateau of wheat reaches at 18, and while this one goes up to 25. This shows that the optimum temperatures is higher. The enzyme, the optimum temperature for maize is higher than the optimum temperature of wheat. So the second point is about optimum optimum temperature. Okay, let's see what the marking schemes say. Marking schemes say measures greater value at all temperatures. Yeah, optimum temperature. Yeah, the same points. We got our 2 marks. Okay. Then we go to the next question, which says that explain the difference between the rates of photosynthesis of wheat and maize at 25 degrees. So at 28 degrees, at 28 degrees, the rate of photosynthesis of maize is much lower. I mean, the wheat is much lower than the maize. This is simply because, now here we should understand one thing, very clear thing, okay, that maize, the rate of photosynthesis declines after 23 degrees because temperature is no longer limiting, okay. But then in wheat it declines after 18 because in wheat over here the enzymes are denatured, okay, and that's why it declines so fast. So that's the point I had in my mind. So maize is a C4 PP carboxylase, more efficient at high temperature than rubisco. So for the respiration occurring in wheat, and the oxygen instead of carbon dioxide combined with RUBP lacks fixation of carbon dioxide, Calvin cycle slows down. So you just had to mention the same point I said, that in maize what we have is actually just a difference. Just the difference here is that wheat is not a C4 while maize is a C4, and maize is less affected by temperature increase, whereas wheat is more affected. And why it's more affected? Because there is photorespiration taking place. And then we can just write down the sequence. That first thing is photorespiration, okay, photorespiration. So now rubisco combines with oxygen, okay, instead of now if you don't understand this thing, take a look at the video on C4 plants, okay. Rubisco plus oxygen instead of carbon dioxide. Fine. And then after this point we go to the next, where we have a photorespiration, rubisco plus oxygen. Okay, then the RUBP declines. Okay, the RUBP declines, and if RUBP declines means the Kelvin cycle will go down as well. Okay, yeah, so this is the light independent stage, will decline. So I hope that point is very clear, because that's what they say here. Text fixation of carbon dioxide, okay? You may wanna add this point over here, that RUBP goes down. So what happens here is that after this lacks carbon dioxide fixation, okay? Lacks carbon dioxide fixation. Okay? There is less carbon dioxide fixation. Hope that was clear, and I'll see you in... No, actually I have another thing to discuss here. But we better finish it here, then we'll discuss the next thing in the next video. Thank you for listening.